Industry analysis shows that over 40% of biotherapeutic drug failures during clinical development are linked to stability issues, with protein aggregation being the primary cause. Does this sound familiar? If you're sitting there thinking that could be us, then this episode could change everything for your company. What if I told you there is a way to build quality in from the start? That's exactly what Quality by Design, or QBD, delivers. Welcome to the Smart Biotech Scientist. I'm David Brühlmann, and today we're diving into something that could literally save your company from becoming another biotech statistic. Let me tell you about a real case that shocked the oncology world. Technopharmasphere highlighted a recent monoclonal antibody drug development case study. Clinical trials were going well, patients were responding, safety looked good, investors were excited. Everything was on track for a successful phase 3 launch. Then came the scale-up. What started as a minor batch inconsistency quickly revealed a catastrophic problem: protein aggregation. Despite extensive upstream optimization, their carefully engineered antibody was forming aggregates during shipping and storage at the very temperatures they'd specified for distribution. The company faced an impossible choice. They could push forward and risk a regulatory rejection or halt everything. They chose to halt production, completely redesign their formulation, and submit entirely new data packages to regulators. The cost? Nearly a year of delays. Production costs increased by over 25%. Their lead over competitors evaporated overnight, all because they'd never properly defined what quality meant at commercial scale. This, unfortunately, isn't an isolated incident. Industry analysis shows that over 40% of biotherapeutic drug failures during clinical development are linked to stability issues, with protein aggregation being the primary cause. Does this sound familiar? If you're sitting there thinking that could be us, then this episode could change everything for your company. What if I told you there is a way to build quality in from the start? That's exactly what Quality by Design, or QBD, delivers. QBD is a systematic approach to pharmaceutical development that begins with predefined objectives and emphasizes product and process understanding based on sound science and quality risk management. As the ICH Q8 R2 guidance states, it's fundamentally different from traditional approaches because it focuses on understanding your product and process deeply rather than just following established recipes. The FDA puts it simply: Quality by Design, and I quote, builds quality into the product instead of testing it. But here is what makes it powerful for biologics specifically. The NIH guidance reminds us that for biologics, the process is the product. This means any changes in manufacturing can fundamentally change your biological molecule. QBD helps you understand and control these relationships from day one. And if I say the process is the product, that's valid for biologics. And we're going to cover biologics in this one. If you're talking cell and gene therapy, actually this is even more critical because the process will change. For instance, if you cultivate in a cell, it can totally change your stem cell, the shape, the characteristics, and so on. So QBD rests on 3 fundamental pillars. First, product understanding, which means knowing exactly what you're building. Second, the quality requirements, which means identifying what matters most for patient safety and efficacy. And third, which we'll cover in our next episode in part 2, process control, because you need to understand how to make your product consistently. The business case is compelling. QBD delivers predictable manufacturing, which reduces your risks. It builds regulatory confidence, which accelerates approval times. And what is the bottom line? Well, it's a faster, cheaper path to market. And there's an additional benefit. You will sleep better. Guaranteed. All right, let's look at the pillar number 1, your product blueprint. Every successful product starts with knowing exactly what you're building. That's where the quality target product profile, or QTPP, comes in. Think of your QTPP as your product's blueprint for success. It links patients' needs directly to manufacturing specifications. The key insight? Well, start with the patient in mind and work backwards. So in other words, start with the end in mind. Your QTPP should define 4 critical components. Number 1, the route of administration. So that means how will patients actually receive this therapy? Is it a pill? Is it injected? Is it freeze-dried or whatever it might be? Second, the dosing regimen. So how often and at what strength? And third, the stability requirements. So what are your storage and shipping constraints? And finally, number 4, the quality expectations. So there we look at what purity and potency targets must you hit. Let me give you a concrete example from the AMAP case study. The industry's gold standard example developed jointly by regulators and industry. This hypothetical monoclonal antibody program demonstrates how a clear QTPP drives every manufacturing decision. Their QTPP specified subcutaneous administration for patient convenience, which immediately drove formulation requirements for high concentration and low viscosity. The stability requirement of 24 months at 2 to 8 degrees shaped their packaging and distribution strategy. Most importantly, their quality expectations, including aggregation below 2% and purity above 95%, became the foundation for all subsequent CQA definition. Here's how to define your own QTPP. Start with the patient perspective. So what do they actually need from your therapy? Consider your clinical requirements. So what safety and efficacy targets must you achieve? Factor in commercial reality. So how will market access and competition affect your positioning? And validate manufacturing feasibility, which translates into, can you actually produce what you're promising? Now let's look at the second pillar, the critical quality attributes, or CQAs. These are quality attributes that directly impact patient safety or efficacy. The ICH Q8 guideline defines them as properties that must be controlled to ensure quality. The focus principle is simple. Identify the critical few, not the many. Not everything that can be measured should be measured. And that's particularly a challenge now in the 21st century when we have so many analytical methods at our disposal. So that's the fundamental principle behind CQAs. Measure what really matters. CQAs typically fall into 3 categories. The first one are all safety critical attributes. They include, for instance, things like aggregates, attributes that create immunogenicity risks, and endotoxins or bioburden that could harm patients. The second category are all efficacy critical attributes that, for instance, include potency, the binding affinity, the biological activity. So all the properties that make your drug work. And finally, the third category are all stability critical attributes, and they include fragmentation, oxidation, and charge variants that affect shelf life and performance. So how do you identify your CQAs? You will start, and this is what I advise my clients, is start with a risk assessment. What could actually hurt patients? Consider your mechanism of action. How does your drug deliver its therapeutic effect? And leverage prior knowledge. What do similar approved products control? and validate analytical capability. So, which basically means, can you measure these attributes reliably and consistently? Let me give you a concrete example from the industry's most celebrated QBD success story. So Roche Genentech obinutuzumab, or the commercial name Gazyva, became the first monoclonal antibody approved by the FDA using comprehensive QBD principles in 2013, including an approved design space. This breakthrough came after learning from their earlier Perjeta submission, which was rejected as a full QBD filing. Here's how they systematically identified their CQAs. During the evaluation process, Genentech discovered that ADCC, antibody-dependent cellular cytotoxicity, had initially not been identified as a CQA based on characterization studies. However, the regulatory review process highlighted ADCC's potential impact on safety, leading to the conclusion of fucosylation patterns as a critical quality attribute, since fucose levels strongly correlate with ADCC activity. The predominant afucosylated form, the G0F, showed strong correlation with ADCC and was added to their CQA list. Their final CQA profile included several key attributes that directly impact the patient: safety and efficacy. Aggregation levels were controlled below specific thresholds due to immunogenicity concerns. They also monitored potency, purity, and specific charge variants that could affect therapeutic performance. This systematic approach delivered transformative business results. According to Genentech, the comprehensive QBD approach provided unprecedented regulatory flexibility, though it required extensive justification and significantly increased the CMC section of their biologics license application. The enhanced process understanding allowed them to make manufacturing changes within their approved design space without additional regulatory approval, providing both operational flexibility and reduced time to market for process improvements. Genentech has since applied these refined QBD tools to their entire biologic pipeline, and thus creating significant efficiencies and resource savings during late-stage development, while enabling post-license flexibility for production. But most importantly, the rigorous CQA identification process ensures that every quality attribute they control has a direct scientific link to patient safety and the therapeutic efficacy. This systematic approach aligns perfectly with the ICH Q8 guidance on pharmaceutical development, which emphasizes that quality should be built into products by design. The guidance specifically states that the product should be designed to meet patients' needs, exactly what the QTPP accomplishes. Now, here is something critical that many founders miss. The FDA and EMA actually want to see QBD approaches in your submissions. They're not just checking boxes, they want evidence that you understand your product and your process. Hence, start engaging with agencies early through pre-IND meetings, present your QTPP and initial CQA thinking, and they'll guide you on what matters most for your specific molecule and indication. If you are working or leading a small company, this early dialogue is gold. It prevents you from over-investing in the wrong areas and shows regulators you're thinking systematically about quality and ultimately patient safety. Before your next team meeting, I want you to do one simple exercise to put today's episode into practice. It's called the CQA reality check. List every quality attribute you currently test for your product. Now ask yourself this binary question for each one. If this attribute failed, would it hurt patients or impact efficacy? If the answer is yes, it's a CQA candidate. If the answer is no, it's just a nice to know. Your goal is to narrow your list to 5 to 8 true CQAs in this exercise. Focus on what actually matters for patients, not what's easy to measure. This single exercise will give you the foundation everything else builds on. You'll stop overtesting irrelevant parameters and start focusing your resources on what truly drives product quality. Before we wrap up today, I want to address something I hear constantly from founders. David, this all makes sense, but how do I make sure I don't miss any critical steps on my path to IND filing? Well, very good question. Here's the reality. Most biotech founders spend 2 to 4 weeks just mapping their CMC roadmap. Meanwhile, every successful monoclonal antibody program follows the same proven path to IND submission. And that's why I've developed the Notion CMC dashboard, a comprehensive digital workspace that eliminates the guesswork from your CMC journey. This isn't just another Excel template, it's your CMC command center that transforms complex roadmaps into an intuitive system that successful programs rely on. The dashboard includes a complete proven monoclonal antibody CMC roadmap to IND submission, multiple views including your tasks, the key activities, the timeline, and the risk and delay alerts. Plus, it's fully customizable to your specific needs. No setup required, it's ready to go. This dashboard transforms your startup's CMC journey from chaos to confidence, giving you predictable execution instead of hoping you didn't miss a critical dependency. But here's what really gets founders excited about this solution, the visibility. Notion's powerful database views lets you see your entire CMC program at a glance. You have Kanban boards that show task flow, you have timeline views that reveal critical path bottlenecks, and you have also an automated risk alert flag potential or a flag system that shows you the delays before they derail your IND submission. As an added benefit, your investors love the transparency and your team stays aligned and you sleep better knowing nothing's failing through the cracks. So think of it as your CMC blueprint that actually works. Not just another static document, but a living system that evolves with your program. If this is something that could simplify your CMC development, check the show notes for details on how to access the Notion CMC dashboard. And also you can there watch a 5-minute demo video and you can let me know if this is something that is of benefit. So as we're wrapping up today, so here you have the foundation. You know what you're building through your QTPP and what quality attributes matter most through your CQAs. In the next episode, we're covering pillar number 3, how to control your manufacturing process to consistently hit these targets. Plus, I'll reveal how hybrid modeling can help you reduce experiments by up to 80% while getting better results. The techniques we'll cover could save you months of development time and millions in unnecessary experiments, so don't miss it. This has been the Smart Biotech Scientist Podcast. I'm David Brühlmann, your host. Thank you so much for joining us, for tuning in today. And please leave us a review on Apple Podcasts or whatever platform you found us on. This will mean the world to me. And I'll see you next time. Thank you so much and see you soon. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.